Hey guys, I miss you all. I wish we could finish off the school year strong in person, but that's just not how it's going to be. But I do want you to know that I miss you and I love you, and I hope that you're okay. If you're not, please reach out to me. I'm always here for you. Okay, so this lecture is going to be probably about 4 to 5 days of notes, so do not watch all of it at once. It'll be overwhelming. You'll probably watch 5 to 10-minute intervals based on what I assign you. So do not feel like you need to watch everything at once. If you have your notes, we're going to start on the second page where it says evidence of evolution. Number 1 is fossils. So that's where we are on the notes if you have that. And let's go ahead and get started. So if you remember back from the natural selection PowerPoint, we talked about evolution and how it's a gradual change that occurs over a long period of time. What that means is that it doesn't happen over a day, it happens over many years, and it happens to a whole population. So evolution doesn't occur within an individual, it occurs within a population. Now, evolution is a theory, but in terms of science, a theory isn't something that someone just came up with off the top of their head and there's been no evidence. A theory in science is something that has been substantiated or backed up by multiple experiments, multiple sources of evidence, etc., etc. So evolution is a theory, yes, but that doesn't mean it's not credited. It means that there's a lot of evidence supporting it. Okay. So the first one we're going to talk about, there's 4 main sources of evidence for evolution. The first one is going to be fossils. This is the one you're probably most familiar with because in 7th grade they tend to spend a lot of time talking about fossils. So fossils provide evidence of organisms that lived a long time ago. And so this is really cool because we get to see organisms that don't exist now on Earth, but did at some point. And so through examining fossil evidence, we can make a rough history of life on Earth. Now, it's important to note that it is a very rough history. It's definitely not complete. And that's because most organisms are not fossilized. They may not be fossilized because they don't have hard shells or hard backbones that will fossilize. There may not be enough of them to be fossilized. And so we're probably missing about 90% of the organisms that lived on Earth in terms of the fossil record. So it's definitely not complete, but it does allow us to create a history of life on Earth in terms of really, really long, like 400 million years ago, 500 million years ago, things like that. And a little fun fact is that fossils are found in sedimentary rock. And if you look at the layers of rock, the layer at the bottom is the oldest. And then as new layers are deposited, you're going to get newer and newer organisms. So that's how we can kind of trace the development of life on Earth. Okay, so that's the first source of evolution. The second source is going to be their body structures. What are their bodies like? Do they have similarities in terms of the bones that make up their bodies, the muscles that make up the bodies? Basically, the anatomy or the structures of the organisms. So there's 3 types of these. The first one is homologous structures. If you remember back, homologous means same, or homo means same. And so these are ones that they have the same structure, but they may have different functions. So what that means is they have the same bones that make them up, but they may be used for different things. So if you look at the picture provided, and it's on the PowerPoint, so I'll go ahead and just underline what I'm talking about. We have the human arm, a horse leg, and then a bat wing. Okay? If you notice, they all have a brown bone, a white bone, and yellow bones. Now, something to note is they're not actually different colors in your bodies. This is just to let you see the difference in the bones and what the similarities are. And so since all 3 of these have the same structure, do they have the same function? Well, a human arm is used to pick things up, maybe used to write your science notes when you're learning about biology because it's your favorite subject. The horse's leg is used to move around, and the bat's wing is used to fly. So all of these are used for different things. They have different functions. However, they have the same bones that make them up. So what that tells us is that they share a common ancestor. Very important to note that homologous structures will tell you, hey, these organisms share a common ancestor because they have the same bone structure. So homologous structures, you need to know they common ancestor. So it means that they share a common ancestor because they have the same bone structure. However, they may have different functions. So the function of a human arm is not the same thing as the function of a bat's wing. However, they have the same bones that make them up, so we call that homologous. Second one is analogous. So these are similar in function, but have different structures. So analogous is basically the opposite of homologous. So with analogous, they are used for the same thing. So we have a shark fin, a penguin's flipper, and a dolphin's fin. Okay, all of those are used to propel through the water. But if you look at the bone structure of each, and you have it in your notes, you have it on this screen right here, if you look at the bone structure, they're incredibly different. That tells you that they do not share a common ancestry. Okay, so homologous structures say, hey, they share a common ancestor, they're pretty closely related. Analogous structures say they both have the structure used to do the same thing, but the bones are completely different, so they don't have a common ancestor. So we would say that dolphins and sharks do not share a common ancestor or a recent common ancestor because their bone structure is so different in their fins. Okay. And then lastly, we have vestigial structures. These are my favorite. They're kind of really cool to me. So these are going to be structures that are just leftovers. They're not used in present day organisms, but they were in that organism's history. So there's a couple examples in humans. There's the appendix and then wisdom teeth. So with the appendix, your body does not require the appendix. However, if your appendix gets infected or if it bursts, that's incredibly dangerous. So that's the structure that we just kind of have because it was used in the past with our ancestors, but we don't really use it now. Another example would be wisdom teeth. So back in early human history, we were scavengers or hunter-gatherers, and so we would eat a lot of things like nuts and meat, and so we would need those teeth back there to chew up our food. However, as we've become more developed and we have things like agriculture and processed foods, you don't need these teeth in the back. And so what we're seeing is actually the human population is shifting towards not having wisdom teeth. And so that's kind of one really cool present day evolution that's occurring in humans. So that's really cool. And then the third example is an ostrich's wings. Ostriches can't fly, but they still have wings. That's because their ancestors could. And so they still have that trait even though it's not used. It's just considered a leftover. It's not used. Has a reduced function. Okay. Third source of evidence for evolution is embryology. So this is the study of embryos and how they develop. And what we know from this is if organisms have similarities in their embryological development, they're more closely related. So if their embryos develop very similarly, that means they're closely related. If their embryos do not develop similarly, that means they're probably not closely related. So you have this picture right here in your notes. And it is important, if you look at that first row, and I told you one of these is a fish, one of these is a human, you could not tell me which one was which. Okay, that's a source of evidence for a common ancestry between all organisms. So saying that we may be descended from one organism and we are very similar in terms of embryological development at the beginning because of that ancestry. However, if you keep going in the embryological development, so if you go from row 1 or column 1, row 1, row 2 to row 3, you do start to see those similarities, and you probably at the end be able to tell me, hey, this is the fish, this is the human. Okay, so that tells you, hey, the fish and the human probably are not closely related, which is true. But if you look here, here's the little baby human, and then here, right here, we have a pig. So at the third stage, it gives you I don't have corona chill. At the third stage, it is still kind of easy to see, hey, this is a human. But if you look to the left, that's a pig. And so pigs and humans have very similar stages of development. So we would say that they are really closely related. If you look at the human, which is this one right here, and then you look at the chicken, yeah, not so much. They're not really super closely related. And so that's because, I mean, that's a whole different classification. Humans would be mammals, and then chickens would be birds, so that's kind of different. But if you do notice that the humans do develop similarly to the mammals that are listed. So the mammals that are listed are going to be, and it has that under the picture on your notes if you want to read along as well. So we have the human. Oh, I'm sorry, that was a rabbit. My bad. So we develop similarly to the rabbit, not a pig. Oops. Rabbit, calf, and hog. And so those would be the 4 on the right, and those would all be mammals. And so that's why we do see that similarity between development, because they are more closely related. As you go to the left, we have a chick, a tortoise, a salamander, and a fish. And so those are going to be less closely related to the mammals on the right. And then lastly, the last source of evidence, and in my opinion, the most important, and usually in science's opinion, the most important, is molecular evidence, aka DNA. So the more closely related organisms are, the more similar their DNA is. And you know this. You know that you have half the DNA from your mom, half the DNA from your dad. So if I were to do a test, you would have the most similarity of your DNA probably to your parents or your siblings. If you have an identical twin, obviously you're going to have the same DNA. So the more closely related organisms are, the more similar the DNA is. So with evidence in terms of evolution and studying evolution, what we do is we sequence an organism's genome, that's all their genes, all of their DNA, and see how many similarities there are to another organism. If there's a lot of similarities, that means they're more closely related. If there's less similarities, that means they're less closely related. It is important to note, if you remember, we use DNA to make RNA to then make a protein. So with the DNA, we're actually going to be looking at the amino acid sequences. So the more amino acid sequences they have in common, the more closely related they are. The less amino acid sequences they have in common, the less closely related they are. Okay, so that's the 4 sources of evolution, and we're almost done. We just have a little bit more to get to. And so I just want to introduce these terms so you are aware. Camouflage, you're probably already aware of what this is, but just so you have the scientific definition, this allows organisms to become almost invisible to predators. So this is what we call an adaptation, something that helps an organism survive. If they're able to blend into the environment, they're less likely to be seen, and they're less likely to be preyed upon. So that would increase their chance of surviving and reproducing, and so that would make them more fit. So that's camouflage. And the other term you need to know is what we call mimicry. If you're mimicking someone, that just means you're copying them. And that's basically what mimicry is. So it's when one species is going to evolve to look like another species. And the reason why they do that is because the species they're evolving to look like is dangerous. And so this is genius. This is one of my favorite all-time things that organisms do in the environment in terms of evolution. And here's a really good example. So you have the monarch butterfly right here, and then you have the viceroy butterfly right here. The monarch butterfly is actually extremely toxic to birds, and so they will stay away from the monarch butterfly. They will not eat them, they will not go near them, because they know if they eat the monarch butterfly, that is immediate death. The viceroy butterfly actually tastes really good to birds, and it doesn't kill them. It provides them with nutrients, and it's a good snack. Well, what we've seen is over time, the viceroy butterfly has actually started evolving to look like the monarch butterfly. And it does that because if it looks like something that's scary or something that's poisonous or something that's super predatory, then it's less likely to be eaten, meaning it's more likely to survive and reproduce. So this would be another adaptation we see in nature. Another example is the coral snake and the king snake. I can tell you if I was out, I would stay away from both. I don't care if it's venomous or not. That's not Ms. Hall's forte. Snakes are not her thing. But just for you to know, the eastern coral snake is venomous. The scarlet king snake is non-venomous. And they look close enough to where I would stay away from both. Okay. And so something you do need to know is what adaptation is and the exact definition. So it's just inherited characteristics that increase an organism's chance of survival. So anything that you can pass on to your children that has increased your chance of survival, meaning it has to be genetic. So camouflage would have to be genetic. Something in your genes causes you to look like the environment. Mimicry, something in your genes causes you to look like another organism. That's dangerous. And so adaptations can be physical, which is what we've talked about, or they can actually be behavioral. So in terms of an organism's behavior, something that increases their chance of survival because of their behavior. So some examples of that would be bird calls, mating dances, and migration would all be different behavioral adaptations. Okay, so this is the end of day 1. I love you guys. Have a good day. This is the beginning of day 2's notes, so welcome back. We're going to talk about the types of natural selection. So the first type of natural selection is what we're going to get into, but just to review, natural selection is when individuals that are best suited to their environment are going to be selected for. What that means is if they're more likely to survive and reproduce, they're going to be selected for in terms of natural selection. Now, it does depend on the environment, and as the environment changes, the ideal fitness will change as well. So if we remember back to the rock pocket mouse video, when the terrain was a light brown, you had a large population of light brown mice and very little black mice because it would be really easy to see a black mouse on sand, and so the hawks could easily go for that black mice, removing those genes from the population. As the lava flow erupted and the environment became dark, we saw less light brown mice and more black mice, and that makes sense because it's very easy to see a light brown mouse on black surface, and so we would remove those genes from the population by the hawks eating them. That's kind of how that happens. Okay, so there's going to be 3 types of natural selection. The first one, just so you know, we are right here. You should see these 3 types. So this is where we're at on the notes. The first type is stabilizing selection. So if you're stabilizing something, you're making it steady. And that's kind of what stabilizing selection does. So it eliminates extreme phenotypes. What that means is if you have, and it does this in human height, so if you have very short humans and very tall humans, they're not common in the population. And the reason why is because there's health side effects to being very tall or very short. And so with human height, we do see a stabilizing selection, meaning that the extreme phenotypes, super tall, super short, are not selected for. They do not increase an organism's fitness. And so we will see that average height, it does increase fitness and helps a human survive and reproduce. Another example would be the Siberian Husky. They have a medium height and build because what they do is they travel through snow. So if they're too tall, they cannot travel through the snow correctly, and if they're too short, they cannot travel through the snow correctly. So what we're going to see is most Huskies are going to be in the middle. They're going to be the medium sized because that is where they're most fit for their environment. Second type is directional selection. So this goes in one direction. So it favors one extreme. It doesn't favor the average, it favors one of the extremes. So again, if we had small, medium, and tall, if we had directional selection, it would favor either the tall or the short. It would not favor the medium. So with a real world example, this would be like greyhound dogs. So greyhound dogs are very large. They're definitely on the tall end, and most of their population is much more taller than the average dog population. And so the reason for that is because they are bred for speed. So as they get taller, they get faster, generally. And so we would see that instead of it being down the middle with greyhound dogs, as with the Huskies, greyhound dogs are actually much taller. So that's going to be directional selection. It's shifting towards one of the extreme phenotypes. And then lastly, we have disruptive selection. So this is when you're splitting a population into 2 groups, and you're favoring actually both of those extreme phenotypes, and the average phenotype is not favored. So you will not see many organisms with that average phenotype. So a real world example is if you had an environment with both black and white rocks, you would see a lot of black mice, or I guess we're using rabbits here. You would see a lot of black rabbits and a lot of white rabbits. The white rabbits could blend in with the white rocks, the black rabbits could blend in with the black rocks. You're not going to see many gray rabbits because gray does not blend in with black and does not blend in with white. And so you would see large white population, large black population, and almost no gray. That's what we call disruptive selection. So you're going to have two peaks in a disruptive selection graph. Okay. Gene pool, so you can literally think of it as a swimming pool for your genes in a population. So it's just all the genetic info of the members of a population. So it contains the alleles, you know that already from Mendelian genetics. And another term you need to know is the allelic frequency. So this is the number of times an allele occurs in a population, and we're going to measure it in a percentage. So an example would be you have 70% big A in a population, 30% little a. So that means 70% of the organisms have the big A allele, 30% of the organisms have the little a allele. That's how you would calculate allelic frequency. And then lastly, we're going to talk about Hardy-Weinberg for today. You don't need to memorize anything on this, but it is really cool, and it is a very important topic in terms of evolutionary science. So the Hardy-Weinberg principle says that when allelic frequencies stay the same, meaning that if you have 70% big A, that does not change. So over 10 years, you'll still have 70% big A. So if those allelic frequencies are staying the same, a population is going to be in genetic equilibrium, meaning the population is not changing and is not evolving. So the Hardy-Weinberg principle is how we measure how much a population evolves. In nature, all populations are evolving all the time. So what we need to determine is how much are they evolving. And this does depend on certain factors, and we're going to talk about those in the next slide. Okay, so there are really 5 factors that determine how much a population is evolving. So in order for there to be no evolution, all 5 of these conditions have to be met. You have to have a large population. You have to have no immigration, which is just movement of genes. You have to have random mating, meaning that mating cannot be selected for based on desired characteristics. You would have to have no mutations occurring. Mutations are random, so this never happens in nature, but that's why we have evolution constantly occurring, because we always have those mutations just randomly happening. And then lastly, you would have to have no natural selection. So in nature, this does not happen. So this allows us to see how much a population is evolving. Okay, so that is it for today's notes. Have a good day. I miss you guys. And if you have questions, please reach out and email me. Okay, so today we are on day 3 of the notes. We are right here where it says other factors leading to evolution. And there's going to be 3 things that we're talking about today: genetic drift, gene flow, and sexual selection. Okay, so what is genetic drift? If something is drifting, think of street racers when they drift their car, they're kind of moving. That's what genetic drift is. What it's saying is that the genes are moving in a population. They may be coming in, they may be leaving, but for some reason the allelic frequencies are changing. This is due to random mating and chance, so just random. Genetic drift is going to be much more pronounced, and you're going to see a larger effect of genetic drift in small populations. Because with small populations, if you have an organism die off and that organism is the only person who has a little a in that population, you now have no little a's in that population. So genetic drift is much more pronounced and evident and obvious in small populations. And I did give you this picture on your notes. If you notice, we have 3 red frogs. If these 3 red frogs die off, we now have no red phenotype in the new population. And so because the initial population of frogs was so small, we only have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I can't do math. like a small number, then when those red frogs die off, we have no more red frogs. So we've lost those alleles permanently from the population. And so genetic drift definitely causes a loss of genetic diversity in those small populations. And so this is why you see, if you have isolated populations, you tend to see that they have very similar DNA, they have very similar alleles, because they have no one else to mate with and they're losing that genetic diversity. Okay, so there's really two types of genetic drifts we're going to talk about. The first one is the founder effect. So if you think of the founder of the United States as someone who traveled from Europe and came and colonized the United States. So that you can think of that as kind of what the founder effect is. So it's when organisms from one population leave and establish a new population somewhere else. And so if you look at this island right here, we have a bunch of butterflies on the original island. And then let's say 3 butterflies leave and colonize a new island. On the original island, I see 3 phenotypes. We have orange butterflies, black butterflies, and white butterflies. On the new island, we only have orange butterflies and black butterflies. So through the founder effect in this example, we've eliminated a whole phenotype. And so the founder effect definitely causes a loss in genetic variation because you have to depend on whatever your organisms have in terms of their genes. So if they don't have the white allele, then you're not going to have any white butterflies. And so again, the founder effect is just when a new population is established or made by a small number of individuals from a larger population. And so this always, well, I don't want to say always because that's pretty definite, but 99.9% of the time will reduce your genetic diversity. The second type of genetic drift is going to be a bottleneck effect. And so these are going to be caused by natural disasters most of the time. So things like flooding, tornadoes, drought, anything that would kill off a large population or a large percentage of a population is going to cause a bottleneck. And so if you look at the 2 bottles in this picture, the one on the left and the one on the right, the one on the left is primarily yellow. So there's mostly yellow in there. The one on the right is mostly blue. And so what a bottleneck effect does is it changes the genetic makeup of a population. And that's because it only allows a few individuals to survive. So a lot of individuals are killed off because if we have a drought, they can't eat anything. Well, those few that survive will then reproduce and create a new population. That new population will not resemble the old population, most likely because those few individuals that survived are not representative of the population. They just survived maybe randomly, maybe due to something that they had, etc., etc. So we see our population shift from mostly yellow to mostly blue, and that's because randomly through the bottleneck effect, we had much more blue organisms survive than yellow, and that's just why that happens. Oh, I forgot to tell you something really cool about the founder effect. So an example of this in the human population in terms of the US, the founder effect, is the Amish population. So the Amish population is very close-centered and closed off. And so because they are isolated in terms of breeding and population, they actually tend to have many more certain genetic disorders that are passed down from generation to generation. So that's kind of something really cool to look into if that interests you at all. Okay, second type or other factor that leads to evolution is gene flow. So this is literally genes flowing in and out of a population. This is also what we call migration. So there's 2 types. You have immigration with an E. This is when genes are leaving or exiting. So that's how I remember immigration with an E, genes exit. Immigration with an I, genes come in, because in starts with an I, exit starts with an E. So that's kind of an easy way that I use to remember which one means which. If you do not have gene flow, you're most likely going to have the 2 populations evolving into different species. So this is what we saw with Darwin's finches. the finches would travel to new islands. As they traveled to new islands, there was no gene flow. There wasn't any more organisms coming from the main island to the new islands. So since there was not that immigration, you had evolution of different species. And so you had multiple different species of finches on the different islands based on what their environments were. And that's because the islands were so separated, you didn't have that gene flow. You didn't have organisms just coming back and forth all the time and mating and reproducing, things like that. Okay. And then lastly, you have sexual selection. So this occurs when certain traits increase reproductive success. So this one is really interesting because sometimes it's kind of funny, because although they may the traits may increase their reproductive success, they can actually reduce their chance of survival. So prime example would be the male peacock. And the male peacock is the one with the really pretty feathers, but they're so big. And so because those feathers are so large, it's hard for a peacock to move around, a male peacock. So why on earth would evolution have selected for very large feathers if you can't move? Well, it's because the female peacock sees those large feathers and she's like, ooh, he is cute, based on how big his feathers are and how colorful they are. So that's something that does happen with sexual selection a good bit of the time, is it may select for a trait that does not increase its chance for survival, but increases its chance for reproductive success. So because those feathers are so attractive to the female peacock, male peacocks will evolve to have bigger and bigger feathers. Okay, so that is it for today's notes. If you have questions, definitely reach out and email me. I love you guys and I miss you guys. Have a good day. Okay, we are back on day 4. Welcome back. We are going to talk today about, it's basically just the random stuff that I couldn't fit anywhere else. So we're just throwing it all into one presentation. So just being honest. Coevolution is where we're going to start off. So we are right here on the notes. You should see it's right under the peacock, the male peacock with his large, attractive feathers. We are right here. You should see a hummingbird with coevolution. So coevolution is a relationship between two species that is so close they evolve together. So prime example would be a hummingbird's beak and the flower that it consumes, the nectar it consumes. As the hummingbird's beak grew, or as the flower grew in length, the hummingbird's beak then evolved to be bigger over time because it would be able to better access the nectar. And so with coevolution, it's usually going to be organisms that have a mutualistic relationship. So they're both benefiting in some way, so they tend to evolve together. That's coevolution. So evolving together, usually a mutualistic relationship is occurring. Then you have convergent evolution. So convergent evolution is unrelated species actually evolve similar traits. So prime example would be echolocation in bats and whales. They are not closely related at all. However, they both use echolocation. So that's a type of convergent evolution. They both evolved echolocation even though they're not closely related and they live in completely different parts of the world. And so that's convergent evolution. And then you have divergent evolution. When things diverge, they split. And so this is when closely related species evolve different traits. Prime example, Darwin's finches. So when they had to adapt to their new environments, they evolved different beaks over time. So that's definitely divergent evolution. They evolved different traits because they each had different environments. Even though they're closely related, they all evolved from one species. They had to evolve different traits in order to survive because they all had different environments. Okay. Prezygotic isolation is really cool to me. So pre means before, and prezygotic isolation basically prevents reproduction for some reason or another. So there's 5 types we're going to talk about. So 5 types of prezygotic isolation mechanisms, and each of these will prevent reproduction from actually occurring. So the first type is behavioral. With behavioral isolation, something with their behavior causes them not to mate with each other. So they may have different patterns of courtship. Certain species of frogs actually do a hand waving thing. And so what they do is the male will see the female and he'll wave his hand. If the female says, that hand wave wasn't good enough, you're not that cute, like your hand waving is just not doing it for me, then they don't mate. So they may have some type of behavioral isolation that prevents them from mating. Temporal isolation. Temporal means time. So something going on with the timing of their reproductive systems will prevent them from mating. So if they're not reproductively mature at the same season or month or year, then they will not mate with each other. You have geographical, aka habitat isolation. Basically, there is a physical barrier preventing mating from occurring. So there's a canyon in the way, there's a stream in the way, they're in different parts of the world. Anything like that would be geographical isolation. Mechanical isolation, literally what it looks like and what you think it is. The genitalia do not fit together for some reason or another. They have different sizes, shapes, or different locations, and so the genitalia are not able to reproduce. And then lastly, you have gametic isolation, so something with the gametes. The gametes cannot be fertilized by each other, so for some reason or another, they're not compatible, so the sperm cannot fertilize the egg, something like that. Okay, so that's prezygotic isolation, so things that prevent species from mating. Then we have postzygotic isolation, which is when the species were able to mate, but their offspring are not fertile, meaning that their offspring cannot have any babies. So prime example of this would be a liger. So if you have a lion and a tiger, they can actually mate even though they're different species, and they do produce an offspring. They produce a liger, and that's a picture of it right there. However, that liger, even when mated with another liger, cannot make babies because it is sterile, meaning that it does not have the gametic abilities. Its gametes do not work correctly, basically. And so postzygotic isolation does not allow the offspring to make more offspring. Another example would be if you mate a horse and a donkey, you make a mule, but that mule is sterile, so even if you try to mate that mule with another mule, you cannot make that. Okay, so that's postzygotic isolation. The egg and the sperm have fertilized each other. You may even develop an embryo or a baby, but that baby cannot continue that lineage. The baby cannot have babies of its own. So that's postzygotic isolation. Okay. And then lastly, we have artificial selection, which is also called selective breeding. This is when humans get involved. We've been doing artificial selection for thousands of years since agriculture started. So if you've ever grown a plant, which most of you haven't, shout out to the student I had who thought broccoli was a tree. You know who you are. If you grow plants, and let's say you're growing corn and you see this one corn plant has tons of corn on it, but what you're going to do is you're going to take seeds from that plant and plant them next year. That's artificial selection. So when humans are intentionally reproducing organisms based on their desirable traits. So prime example would be crops. If you have a certain plant that produces a lot of food, you're going to take the seeds from that and plant them the next year or the next growing season. Another example would be dog breeds. So dogs actually were 100% created by humans. They evolved from the wolf, and humans have created all the different breeds. And so that's definitely a form of artificial selection. It's just humans getting involved with the evolution of organisms. And this happens all the time because, as I've said so many times, humans are just the best species on Earth, and we get involved in everything. So artificial selection is very common. Okay, that is the end of this PowerPoint. If you have any questions, please feel free to reach out. I love you guys. Have a good day.